Hello everybody, welcome back to our channel. Today is biology again. Okay, we talk about protein, cell and nature, but very funny, interesting topics. When I prepare, we kept a lot of things too. So first of all, we need to understand protein is made up of amino acid and how to recognize amino acid. Amino acid has a specific feature. It has NH2, amine group, and a carboxyl group, COOH, and also an R group and hydrogen. And all these four things are bonded with one carbon atom, and we call it an amino acid. Amino acid. Of course, later on we can accept amino acid to have more than the R group. We'll talk about it later to conjugate amino acid. And this R group is hydrogen carbon. Okay, only hydrogen and carbon atom are there. Okay. And then you will see how do the amino acid join together to form the protein. You see it had two sides, right? The carboxyl group has OH and the amine group has a hydrogen. Oh, perfect. Okay, this OH and H can join together to form a water. So condensation reaction can happen. Okay, so join together becomes C double bond O and then NH bond together, and we call it a peptide bond. Okay. A beta bond can be formed between amino acids by condensation reaction. Very familiar reaction, right? Yeah, they happen in glucose. Yeah, monosaccharides and also glycerol and the fatty acid string, right? So a dipeptide is formed. And this is a very interesting because you see, after forming peptide bonds, still have rooms to form the peptide bonds, right? So this formation of peptide form can continue and then they can form a polypeptide. Yes, the condensation reaction can continue and then a polypeptide is formed, a linear chain. And then you look at here, every part has different R groups, right? Different amino acid, okay? So they have an infinite combination of amino acid can be formed by this simple linear chain of structure. We call it primary structure. Okay. So we need to join together. Remember condensation. There is hydrolysis, the opposite, right? I remember for the monosaccharides we can decompose the disaccharides to be monosaccharide by hydrolysis. Triglyceride can be dehydrolysis to go back to glycerol and free fatty acid. Also for the dipeptide and polypeptide, we add the water there and then we can do the hydrolysis and go back. But all this thing doesn't happen by itself, of course. Right? This kind of catabolic and anabolic reaction, okay, they don't appear by themselves. So we need someone to help it. Who are they? Enzyme. So specific enzyme have the formation or decomposition of them. Okay. And we have the word equation, although we learned it before, but we kept it here, okay? Reactant plus reactant but reactant, and then long arrow talking about the reaction type, and then what do you need, and then pick up product and product, okay? This is the formula of a chemical reaction. We learned it before, okay? But now we learn it in details, okay? And then the catalyst and specific conditions are written on the arrow, so the arrow can be long. If there's nothing to describe, then short the arrow. If you want to describe something at here, for example, you can say hydrolysis, you can say enzyme here, maybe some specific requirement. What's the temperature, what's the acidity, or whatever. So for the condensation, two things joined together become a big thing, right? And the water is released. Hydrolysis, a big thing, add the water to it to break it down into two small things. Essential and non-essential amino acid. Essential amino acid cannot be synthesized and must be obtained from food, okay? In other words, a diet must contain this essential amino acid, okay? For example, blah, 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 blah. You don't need to remember them, just remember that in the world some amino acid cannot be formed by the other amino acid. We call it essential. And some non-essential amino acid, always a bigger one, okay? And they can be formed by other amino acid, are not. So you have this concept, okay, essential and non-essential, because they can be formed by other amino acid or not. Okay, it is the how to judge it. Okay, and for the workings, okay, if you have the diet, okay, and then you have to be careful, pay a little bit attention on the essential amino acid, okay, but it can be done, okay, there's no problem about it, okay. For example, some license is especially absent, okay, so you have to be careful. And the peptide change, okay, quick variety of the peptide chain, yeah, I know it, okay, every in the peptide chain, the R can be different. How many different R? 20 amino acid, okay. So when you join them together, every R can have a 20 candidate. So you have learned mathematics, right? Well, if you have two joined together, it's 20 square is the combination. If three joined together, 20 cube. Wow, four joined together, 20 to the fourth, okay? Or over the, yeah, okay. So a lot of combination, okay? And then you can see, you see, there can be any number of amino acids to form a peptide change, okay? It can be a few to 1,000. So imagine if it's 1,000 of amino acids joined together. Each amino acid can have 20 candidates, so it's 20 to the power 1,000. Infinite, okay? Even the calculator surrender can't calculate it. So you can say a polypeptide has different number of amino acids in different sequence, so infinite variety, okay? So that's excellent. So protein can be a different form, different structure to have different function. And then because of the bonding, okay, so the pH and the temperature may break the protein. You learned it before, what the nature of the protein. Now we want to describe it more in details, a little bit in detail. For example, the nature by heat, okay. When you heat up the protein's structure drops, okay. Well, not so unison, 100% that kind and then drops, okay. So delature. So delature first concept is not breaking of the peptide bond, not that strong. Just a little bit changing the structure and shape of the protein is function change, okay. So it loses function and then we call it delature already. Not exactly breaking down of the whole protein, okay, so be careful, okay. So maybe because the protein may have out group or non-out group, they have some hydrogen bond between them, so this hydrogen bond is weaker than the covalent bond, thicker than the peptide bond, right? So it will be altered or break by the high temperature, high kinetic energy of the molecule easily, okay, the shape may change, okay, by temperature. Of course, if you have overheating the protein, so high temperature, you can even break the peptide bond into amino acid. And it can't be formed back because we don't have the M side, okay? And the deletion by the acid and base, a protein can have a polar bar, protein have some parts that has acidic or basic, okay? So this environment of the acidity and the basicity of the environment may alter the shape of the protein, the 3D structure. We always have some pH that is optimal for specific protein. Deviate from that, the hydrogen bond of the protein that may be broken and also may change the 3D structure and then it may be denatured. So the same concept, remember, not breaking the protein. Change of its structure is enough to denature it, to, uh, to, to force it to lose its function. Additional higher level, okay, SL finish. Now it's additional higher level. Okay, first of all, we talk about the diversity in the R groups. Okay, R groups amino acids have a huge chemical diversity. Remember, you have 20. First of all, they can be hydrophobic or hydrophilic. Okay, they can be charged, polar, acidic, or basic. So we have a lot of discussion about this kind of R groups and this kind of property of the different protein. And then after all this, bring a 3D conformation of the protein. Okay, three-dimensional. Okay, so the genes and the protein synthesis. Okay. And then the precise position of the amino acid control the three-dimensional structure of the protein, right? And the protein for the specific function has to have a precise, predictable, repeatable structure, okay? So how can, who the manual, okay? How to connect them, okay? Which, after which, who load it? The DNA load it, okay? DNA, the code for this amino acid sequence is in the DNA. Oh, I got it. So DNA is important. DNA controls what kind of protein to be synthesized, and then the protein has its specific function. Okay, And on each code DNA, there's different codon. Okay, 3 codons that control which amino acid to be put in. So 3 codon. Remember, each codon has 4, right? So altogether has 64 combination, and some are repeats. Okay, some different codons, 3 codons, will synthesize the same amino acid. So some overlap, not 64 different amino acid. Okay. So amino acid sequence is controlled by this 3 codon. Okay. And the 3D conformation and the structure protein is determined by 2. And then universal genetic code, we find it very funny. Every creature on the earth use the same structure of DNA, RNA, okay? And then all of them use the same coding system, okay? The 3 codon, this 3 codon synthesize that amino acid, put the amino acid inside. Every creature use the same code, okay? So we think here's the evidence that we have the same origin, okay? Maybe we go to the other planet finding other life, okay? Maybe there, surely we expect to have the different genetic coding. But on the Earth, all of us share the same thing. So we think that we have the same origin of all the organisms on the Earth. And we call it universal genetic code. And then as we talk about the 4 structure of protein, a very funny, interesting system. Primary structure, secondary structure, tertiary, quaternary structure, okay? Building from one to the other, okay? So we talk about the first is the primary structure in protein, very simple, okay? Linear chain, okay? The polypeptide chain does not form bond by itself, okay? It is a linear one. They don't form any bond for each part. And then, of course, they can form bonding with the other polypeptide chain, but forming the hydrogen bond, okay? We still call it a primary structure. Primary structure is a simple chain, okay? And atoms along the polypeptide does not form bond with each other. Very simple, linear chain. Okay? What happens if they form bonding with each other? Yeah, DNA structure. Yeah, helical. Yeah, alpha helix, right? They form a hydrogen bond with each other, so they curl up. Or beta bridge, okay, they just turn back, okay, so they form hydrogen bond, okay, with each other, and then we call it a secondary structure, okay, and it can be an alpha helix or beta bridge structure, and all this thing is the hydrogen bond between them, okay. For example, is this alpha helix, okay, hydrogen bond, turn back, hydrogen bond, turn back, okay, all these can have a lot of amino acid, okay. Left and right, okay. But the alpha plate is just one way, okay. Go back, and that's it, okay. So they're continuous hydrogen bond or just a few hydrogen bond and turn back. So alpha helix and then a beta plate. And all the hydrogen bond is the CO and the NH hydrogen bond, okay. Not from the R group, okay. And then tertiary structure, R group appears, okay. So more variety, okay. But the tertiary structure is the 3D folding of the structure, and the R group appears, okay. R group in the same polypeptide forming different bonding because it's R group, so it can have different bonding. It can be a hydrogen bond, simple one. Ionic bonds, oh, they are charged. Disulfide covalent bonds and the hydrophobic interaction between the R groups, okay. And then why they are charged? Remember COOH, okay. Releasing the H become a negative charge. NH2 absorbing a hydrogen atom become positively charged, so they can be charged and forming ionic bond. The example, just 3 examples you need to remember, is the hydrophobic interaction between the hydrophobic part, non-polar part. You remember hydrophilic thing attract each other. Hydrophobic thing also attract each other. Okay, so they are hydrophobic, they are not polar, non-polar, so they can attract each other. We call it hydrophobic interaction. And also remember NH become NH3 plus, and then COOH becomes COO minus, and they can form ionic bond. And the diphosphate bridge, okay, is very common in tertiary structure. But how can this form? So you know, in some R group, they have a SH group, okay. And the SH group, they get the 2 hydrogen can release, okay, and then forming the SS group. And why there is sulfur? Yes, it is not hydrogen carbon. Okay, it's more than hydrogen carbon, and we call it a conjugate protein. Okay, they have not just our group, other group. For example, this SH shape, okay? And this is the formula. You need to remember this one, even in the exam question. They ask you how these two form the disulfide bridge. Remember SH, find the SH, and then take out the HH, and then join the S together. And then draw the correct structure, and you get the marks. It is the tertiary structure, okay? So tertiary structure have three things more, okay? Hydrophobic interaction between the outgroup, ionic bond, and the disulfide bridge between the conjugate parts, okay? And the tertiary structure, wow, they join together. Alpha structure joined together. And then first of all, we need to understand their bonding, okay? Their bonding may be only due to the R group, okay? Or maybe due to the not say something that's not in the R group, okay? There's some varieties, disulfide bond, something like that, okay? We call it conjugated protein. They have some parts of it, okay, that is not R, okay? And for the non-conjugate, they only have the R group, okay? So all the bonding is R group bonding, okay? And this one can, conjugated one even can have more than a non-peptide bonding, okay? So non-conjugated protein only contain amino acid, okay? So all the bond is a peptide bonds, okay? For example, insulin and collagen, okay? A conjugated protein can have chemical groups other than the R group. Oh, so it is an exception. Before we said that it must be like this, but now we're open, be more open-minded. Some protein can have a different group of R, NH2, and COX, can be more variety even, okay? And the tertiary structure of conjugate protein can have a non-peptide bond by the other parts, okay? Hemoglobin, okay, for the heme group, okay? And the chemical groups other than this are called phosphatic groups. The phosphatic group always come with the vitamin, okay? And technology about the strong electron microscope, and now we use AI, okay? You have a good visualization of the protein structure, and just like the one in the cover, okay? First page, okay? And then a protein with quaternary structure have more than one polypeptides, okay? Each of the polypeptides has the tertiary structure. The other groupings method is look at the shape of the quaternary structure. Okay, we call it globular and fibrous. Okay, you can see here a sphere shape, we call it globular, and a linear shape. Okay, long one, we call it fibrous. Okay, and then this is maybe a hemoglobin. This is a collagen. Right? Okay. And then for the hemoglobin, for example, okay, hemoglobin is a four tertiary structure joining globular quaternary structure protein. Okay, we got a hemoglobin. Okay, we have four hems. Okay, transfer of oxygen. Okay, and insulin is the two long one. Okay, two long tertiary structures forming bond together, and then with this three strings joined together, which become a collagen. Okay, and both these two are fibrous protein. Okay. And then insulin is soluble in plasma. It's used to transport glucose, and hemoglobin is transport oxygen and carbon dioxide. And collagen is for the connecting tissue in body, as flexible. And the polar and non-polar amino acids, you need to remember some amino acids are polar. You don't need to remember the name, of course. You can get it from internet easily. Just remember some amino acids are polar. Few, just 6, okay? And the other 14 or 15 or 16 when are non-polar, okay? And non-polar, not dissolvable in water, hydrophobic, right? Polar, dissolvable in water and hydrophilic, okay? It is the amino acid property. And then hydrogen bond between the polar amino acid contribute to the tertiary structure, remember? The R group, okay? And but a protein have a hydrophobic amino acid, okay? But if the hydrophobic amino acid are clustered inside the globular surrounded by the hydrophilic one, okay, in the outside, the protein can still soluble in water, okay? Just like a group, outside is hydrophilic, inside is hydrophobic, it still work, okay? It's still hydrophilic. Okay? So integral protein, remember, yeah, the one going through the layers of membrane, okay? It can have hydrophobic part, hydrophilic part, right? Thank you for going through with me, okay? And I'm glad I got the protein, okay? Hope you happy to learn. It's very funny, but okay. And it is also a test for you, okay? You will find it, this structure amazed me. I want to study more. Maybe in university you need to learn synthetic biology or some biochemical stuff, okay? So you can learn as many as possible. You can use the microscope, you can use AI to look at the structure, think about how this structure help it to function in this way or that way, okay? Very fascinating, okay? Is it? Yes, then you love biology. And biology loves you too. Okay, see you for next week, okay? Bye.